Hi everyone, today we're going to talk about microbial metabolism. And we're going to do that because so far in this course we've talked a lot about microbial diversity in terms of counting species and comparing presence and absence and abundances of species in different samples. We've talked about how genomic tools are useful for measuring microbial diversity. And now for the next part of the course, we want to talk more about how microbes are playing important roles in ecosystems. That is, what are the microbes actually doing? Not just their diversity, but what are they actually doing in these ecosystems? And in order to talk about that, we need to talk about microbial metabolism. That's what microbes do. They do metabolism. And this is a famously unpopular thing to lecture about. Microbial metabolism is very different from ours. They're very different than us in terms of their metabolism, and for that reason, it can be difficult to engage with this material. So with that warning, I'm going to do my best. I'm going to start with this sort of textbook intro biology definition of metabolism, which is that it's the combination of catabolism and anabolism. Catabolism being the breakdown of molecules to release energy, and then anabolism is the usage of that energy to make stuff inside the cell. So in our own bodies, we're engaging in catabolism when we break down our food into smaller molecules, ultimately releasing carbon dioxide as a waste product. When we do that, we release energy. That's why we eat food, in order to obtain energy. And then we want that energy so that we can do stuff with it. And the stuff that we do with that energy is anabolism, which is making molecules inside our cells. So this consumes energy, and it makes all of the components of our cells and even the stuff in between our cells, everything in our body, we build through anabolism. We get the energy for anabolism by breaking down food, which is catabolism. These two things together are metabolism. Metabolism, that is metabolism. Okay, meta being, the combination of these two things. So in microbial ecology, we're mostly focused on catabolism. When we talk about microbial metabolism, we're mostly talking about catabolism because one of the really interesting things about anabolism is that it's basically the same in all cells on Earth. All life uses the same anabolic pathways. The pathways that E. coli use to build their lipid membranes and proteins and their DNA and RNA are basically the same pathways that we use. So there isn't a whole bunch of diversity in anabolism, which is one of the fundamental observations of life on Earth that indicates that we are all descended from a common ancestor, that all of our building blocks of our bodies are made with basically the same enzymes and the same pathways, or at least enzymes and pathways where we can see clear evidence for common descent from an original ancestor. Whereas catabolism, the foods that we eat, are very different, and therefore the catabolic pathways that we use to break down those foods are very different, and microbes in particular are diverse even among microbes, not only different from us, but also different from each other. So that's what we're going to talk about. That's what we're going to focus on, is the catabolism. And here's a diagram from an intro biology textbook showing one categorization of the different catabolic strategies of microbes. And the first division we have is phototrophs versus chemotrophs. Phototrophs obviously being organisms that get their energy from sunlight. Chemotrophs being organisms that get their energy from chemicals, that is, food. Normally, when we think of chemotrophs, we also think of heterotrophs, that is, organisms that require organic molecules in the form of their food. And autotrophs are typically phototrophs from our human perspective. Autotrophs being organisms that can make their own organic molecules. So like cyanobacteria, algae, phytoplankton, and land plants. So technically these are photoautotrophs because these are two different concepts. They get energy from sunlight, and they use that energy from sunlight in order to fix CO2 and build it up into their bodies, into their organisms. A lot of bacteria and animals are more accurately chemoheterotrophs because they need to eat their organic molecules, they can't fix CO2, and they're not getting energy from sunlight, they're getting all of their energy from those chemicals. But these are actually two distinct concepts, and that can be made clear by showing that there are examples of photoheterotrophs and also examples of chemoautotrophs. And these categories only have examples among the bacteria and archaea, which is why we often ignore them when we are only looking at the world from our human animal-centric eyes and our green plant-centric eyes. So let's talk about some examples of these to understand how this works. So an example of a photoheterotroph are the archaea that live in the Great Salt Lake. The reason why the north arm of the Great Salt Lake is pink is because of these photoheterotrophic halophilic archaea. They have pigments in their cell membranes called bacteriorhodopsin. It's confusing because they're archaea, but they have molecules called bacteriorhodopsins because this molecule was discovered before it was appreciated that these species were actually archaea, not bacteria. And the pigments absorb light, and when they absorb photons of light, it turns on a proton pump, pumps protons out of the cell, and that proton gradient is utilized to make ATP. So this is really simple, very direct conversion of photons into ATP. And these organisms do not have carbon fixation pathways. They need to eat organic molecules they get from the Great Salt Lake. I think that's mostly like dead brine shrimp, I think is basically what they eat, is the molecules that come from the decomposition of animal biomass, like brine shrimp. But they also get energy from light. So they are phototrophs, because they're using light, and they're heterotrophs because they don't have any carbon fixation pathways. They're getting all of their carbon by eating organic molecules. Photoheterotrophs. This might be the single most common metabolic strategy in the world. It was only discovered relatively recently, but the more we look for it, especially with the help of DNA sequencing techniques, it's been appreciated that not only these weird haloarchaea in the Great Salt Lake, but also perhaps most, at least many species of bacteria in the ocean have some version of these pigments that allow conversion of light into ATP. It might be a very common like bonus energy strategy that you have your normal metabolism that you use to get most of your energy, but in addition, if you're living in a place that has sunlight, why not have some pigments in your cell membranes that also give you a little bit more ATP by absorbing sunlight? It's a relatively new discovery, but it might actually be quite common. So those are photoheterotrophs. That's why photoautotrophs are not the only example of phototrophs, because you can also have photoheterotrophy. Now let's talk about some examples of chemoautotrophy. So you can be autotrophic, you can have a carbon fixation pathway, but if you live at the bottom of the ocean where there's no sunlight, then you can't be a photoautotroph. Instead, you have to be a chemoautotroph. And here's perhaps the single most famous example of a chemoautotroph, which are the bacteria that live in the guts of tube worms. So these are tube worms. They are worms that live near hydrothermal vents on the bottom of the ocean. They like living near the hydrothermal vents, but not too close, so they don't get toasted from being too hot, but close enough where they have access to the funky chemicals coming out of the vents, in particular hydrogen sulfide. They transport the hydrogen sulfide into their bodies. The hydrogen sulfide is used as the energy source for the bacteria living in their bodies to fix carbon dioxide. So the carbon dioxide is also delivered by the tube worm to the bacteria living inside of it. And by coupling the hydrogen sulfide and oxygen, it releases energy. It uses that energy for anabolic pathways, carbon fixation pathways that make organic molecules, and then those organic molecules get passed on to the animal. So this worm is entirely built from anabolic pathways that derive their carbon molecules from the bacteria. The bacteria are providing all of the carbon to the worm. These are microbe-powered worms that can get up to 6 feet tall. And this partnership has worked so well that over time the worms have completely gotten rid of their whole digestive system. They no longer have a mouth. They only have these plumes that absorb the hydrogen sulfide, oxygen, and carbon dioxide, so they don't need a mouth anymore. They also don't need an anus anymore because they have no waste products other than the gases that are given off by the bacteria in their body, which easily diffuse out. So they basically have no digestive system at all anymore. They just have these bags of bacteria. So the bacteria are the chemoautotrophs. They're autotrophic, they're fixing carbon dioxide, they're chemo, they're using chemicals from the hydrothermal vents as their energy source. It's too dark for any sunlight. They make organic molecules, and the animal is still a boring aerobic heterotrophic chemoheterotrophic animal, just like all animals. It's just that it gets its carbon molecules from these chemoautotrophic bacteria. Methanogens are another example of chemoautotrophs. Methanogens make methane from CO2, or at least many methanogens do. Some methanogens are actually heterotrophic, but many methanogens are chemoautotrophic. They fix CO2, and a waste product of that carbon fixation is methane. They are not using light to do this. They don't have pigments to absorb photons. They're getting all of their energy from other chemicals, for example, hydrogen gas. So there we go. There are our 4 basic metabolic strategies among the microbes. Again, our chemoheterotrophs, which include us, as well as many bacteria. Our photoautotrophs, which include the plants and other green things. And then the 2 in the middle are only including bacteria and archaea. Those are photoheterotrophs and chemoautotrophs. Now, this chart is really useful. It provides one way of categorizing all of life on Earth. It does not include everything. So one thing it is leaving out is the concept of being aerobic or anaerobic. And in order to talk about that, we now move to the electron tower, which you may have been exposed to at some point in your academic life. And I bet you were glad when you were done with it and were hoping to never see it again. And if that's true, then I'm sorry, here it is again. I really like the electron tower. I know why some students don't like it. I think it's not taught very well generally. I hope I can teach it better. And more importantly, I hope I can show you how important it is for appreciating the diversity of microbial metabolism. So here we go, electron tower. Things that are really good at donating electrons are on the top. I like to call these electron donors, but if you want to call them reductants, you can do that. Things that are really good at receiving electrons, that is electron acceptors, are on the bottom. So electrons, by nature, prefer to flow from top to bottom. Each stage of electron tower, this is showing what this molecule will be converted to if it gets rid of all of its electrons. So if glucose gives up all of its available electrons to something else, for example oxygen, then it will be converted to CO2. That's what this means. If methane donates all of its electrons completely, then it will be converted to CO2 as well. If hydrogen sulfide gets rid of all of its electrons, it can be converted to either elemental sulfur or sulfate, and so on. Okay, so electron acceptors are on the right, and the good ones are at the bottom. CO2 is technically an electron acceptor, but it's not a very good one because it's up here at the top. CO2 from methane, that potential is a bit better than from glucose, but they're both still at the top. Sulfate is kind of an intermediate electron acceptor, better than CO2, but not as good as the things down here. Okay, so if you're aerobic, you're going to use oxygen as your electron acceptor. That's the definition of aerobic. The definition of anaerobic is that you use something else instead of oxygen as your electron acceptor. So aerobic versus anaerobic is simply drawing a line on the electron tower that separates oxygen from everything else. And that's understandable because oxygen is special. It was not present on the planet until about halfway through the history of life on Earth. For half of the time of life on Earth, the world was anaerobic, and we only had these, probably sulfate and above, as our electron acceptors, maybe iron. Nitrate wasn't really abundant until you also had oxygen. And when oxygen appeared on the planet, it completely changed biodiversity permanently. So I think it's fair that we have these special categories of things that are able to use oxygen as their electron acceptor and things that are not. Anaerobic metabolisms are therefore more ancient, and there's actually a lot of good evidence that aerobic metabolisms have evolved more recently, which makes sense because oxygen only appeared recently. Okay, so back to the electron tower. If you're anaerobic, you're going to use something else besides oxygen. So for example, if you're still heterotrophic, therefore you're eating glucose, and glucose here is just one example of an organic molecule. It could be anything. We typically use glucose as our example because everybody likes sugar. If you are heterotrophic, you're eating some kind of organic molecule like glucose, and if you use nitrate as your electron acceptor, then you are going to get some energy out of that. But you're not going to get as much energy out of it as you would if you were using oxygen as your electron acceptor. So that's what these arrows are showing, that if the electrons don't fall as far, then you will not get as much energy out of them. And that is one reason why oxygen is so special. It's an extremely good electron acceptor, so it provides the maximum potential for obtaining energy from oxidizing any of these molecules. So you would get the most energy by oxidizing glucose, but if you used hydrogen and took the electrons from hydrogen and put them on oxygen, that would also be a lot of energy. Also for methane, sulfide, you could potentially get energy from any of these molecules, even from ammonia, which is not very high up. Ammonia is not an extremely good electron donor, but the arrow from ammonia to oxygen is still going down and to the right, so it is physically, chemically possible. And it was discovered relatively recently that this metabolism is actually quite abundant in the ocean, and it's in fact really important for understanding the ecology of the ocean, as we'll talk about next time. So this is an aerobic metabolism using ammonia as the electron donor. An anaerobic metabolism that starts with hydrogen, which is a great electron donor, but using CO2 as its electron acceptor, that is not going to give you nearly as much energy as going from hydrogen down to oxygen or going from hydrogen down to nitrate. So it's not as much energy, but it is still possible. It's still going down and to the right. It'll still give you some energy, and this is in fact the energy redox couple that methanogens use. They take electrons from hydrogen, put them on CO2, and that is enough energy for methanogenesis to occur. Methanogens often get outcompeted in environments with hydrogen that have additional electron acceptors that are available. For example, sulfate. If there is sulfate around, the methanogens will often get outcompeted by organisms that can use sulfate as their electron acceptor instead of CO2. The electron tower does not specify autotrophic or heterotrophic. So just like the previous chart did not include aerobic or anaerobic, the electron tower does not specify autotrophic or heterotrophic. That's additional information you have to bring into it. But the two things together will tell you the whole story. So these aerobic organisms using ammonia as their electron donor, we happen to know from research, as I mentioned earlier, it's a recent discovery, that these organisms are actually able to support a carbon fixation metabolism with the little bit of energy they're getting out of oxidizing ammonia. And so these are actually aerobic chemoautotrophs because research has shown that they are fixing their own CO2, getting their own food by fixing CO2. And they're doing that, they're getting energy to do that from this coupling of ammonia and oxygen. Now, methanogens are also chemoautotrophs, but they're anaerobic. They're using CO2 as their electron acceptor, as I mentioned, and they are special in their own way because CO2 is not only their electron acceptor, but they are also fixing CO2. So they can use the same molecule as their source of carbon through their carbon fixation pathway and also as their electron acceptor. And that's pretty cool and unique. And one of the reasons why methanogenesis has been proposed to be perhaps the most ancient metabolic pathway, perhaps the original metabolic pathway, because of the simplicity of being able to use CO2 as both the source of carbon and also as the electron acceptor. Okay, but the main point from this slide is how by coupling both the electron tower and the concept of autotrophy and heterotrophy, you can categorize organisms quite specifically. And 4 examples being shown here. Next point I want to make is that this is not just theory. This is not just a useful way to make biologists learn chemistry. It's actually extremely ecologically practical. And one way of showing that is by talking about a column of mud. It could be from the lake or from an ocean. And if that mud is relatively undisturbed and allowed to settle out, then you'll get a really nice redox gradient that is a really nice oxygen gradient where you have a lot of oxygen at the surface where it's exposed to the water. the first thing to be consumed in that column of mud will be oxygen, because oxygen is the best electron acceptor. After all of the oxygen is gone, then the column of mud becomes anaerobic, and nitrite and nitrates become the most available and most favorable electron acceptors. And so you'll get organisms using those. Then if you go deeper, after all of the nitrate is gone, then you'll get organisms that are reducing iron. And then after that, you will get sulfate-reducing bacteria. And then after all the sulfate is gone, then you will finally get methanogens. So this is, for example, one reason why if you want to find the methane in the bottom of a lake, you got to dig through some centimeters of mud in order to get to where the methanogens are, because they're going to be at the very bottom where everything else has been consumed and only CO2 is available as an electron acceptor. And this general concept has been used as a really powerful predictive tool in microbial ecology and being able to discover new kinds of metabolism, as well as to figure out important things about how elements are cycled throughout the ocean and in lakes. And we'll just show many examples of that later on in the course. So it's not just theory. This is, in fact, really how it plays out in nature, that the yummiest stuff gets consumed first, leaving the less yummy stuff behind for other organisms. Okay, so a little bit more of how the electron tower works and how it does not work, with a couple of example questions. So which pair of reductant and oxidant, that is donor and acceptor, would provide more energy for metabolism? Would you get more energy by going from methane to sulfate, this blue arrow, methane being the donor and sulfate being the acceptor, or going from iron to nitrate, iron being the donor and nitrate being the acceptor? Think about it for a second. And the answer is the blue arrow, because the blue arrow is longer. That's as simple as it is. You get more energy because the electrons are falling further down the ladder of the electron tower from methane to sulfate than from iron to nitrate, simply because this arrow is short and this arrow is long. It does not matter where you start or where you end up. So just because you're starting higher or you're starting lower, that doesn't tell you enough. You really need to know how far you're falling. And all it is the distance that you're falling. It doesn't matter if you're starting higher or lower or ending up lower or higher. It's the distance that you fall, whether it's up here or down here. The distance is what matters. It's the differential, the redox differential between your donor and acceptor that matters for how much energy you will get out of it. Okay, second example question. Would it be possible for an organism to make a living by using methane as its source of electrons? So methane as a donor, while breathing CO2, using CO2 as your electron acceptor? And the answer is no, because either you would get zero energy at all, because it's the product of oxidizing methane, so that's a net of zero, or if you're using CO2 up here, then you're going up and to the right, which is the opposite direction that electrons will flow. This is not a favorable reaction. You would require energy to do this. If you wanted to somehow force this to happen, you would not get any energy released from this reaction that you could use to fuel your metabolism. So the answer is no. Okay, so I hope I've made a convincing case that these two things together, the phototrophs versus chemotrophs, autotrophs versus heterotrophs, aerobic versus anaerobic, and then within anaerobic, all of these other electron acceptors besides oxygen, those things together can be used to have a pretty good description of any single species on Earth. That is, you can describe any organism on Earth according to its energy source, photo versus chemo, carbon source, auto versus hetero, electron donor, and electron acceptor. Okay, one word about eukaryotes, and just one slide about them because they're boring. It's one of the most remarkable observations in biology that all eukaryotes are aerobic chemoheterotrophs, and it is as specific as that. They are just one very specific example of all of the possible types of metabolism, the whole domain of eukarya. There are some interesting exceptions, like there are some weird anaerobic eukaryotes, but they're the exception that prove the rule. Those anaerobic eukaryotes are clearly evolved from aerobic ancestors, and plus they're usually just using things like nitrate as their electron acceptor, which nitrate is basically aerobic. It doesn't require any specific metabolic capabilities in order to reduce nitrate usually. It's just kind of like a substitute for oxygen. So in terms of evolution, these are really exceptions that prove the rule that the domain eukarya is an aerobic domain. And furthermore, it's a heterotrophic domain because all of the autotrophs in domain eukarya, that is the plants, things with chloroplasts, the only reason they can fix CO2 is because they have chloroplasts, and chloroplasts evolved from bacteria. So all those metabolic pathways that the plants are using to fix CO2, those are bacterial metabolic pathways. They were invented by bacteria. And so again, that's an exception that proves the rule that eukaryotes are fundamentally aerobic and they are fundamentally heterotrophic. Okay, let's go back to this diagram about catabolism, anabolism, and metabolism, and highlighting how the typical way those concepts are conveyed are very particular to aerobic chemoheterotrophic life, that is eukaryotic life. And if we kind of edit this figure to make it more general for bacteria and archaea as well, then we would highlight that oxygen coming in here in this part of catabolism is not the only possible electron acceptor, that you can also have additional electron acceptors at this stage of catabolism. And similarly, at this point, where we have food molecules coming in here, well, if you are an autotroph, then instead of these food molecules, you would be using other electron donors like hydrogen or methane or sulfide. And in that way, this diagram still works for bacteria and archaea. We just need to make it slightly more generalizable and not specific to aerobic chemoheterotrophs. Okay, just a few more points. Here are some examples of organisms with different electron donors, different electron acceptors, and different waste products. If you just want to see some examples of how the electron tower concepts play out with concrete examples, there you go. And then finally, I want to mention a really cool example of a single organism that can be all of these things. The general category for these are the purple non-sulfur bacteria, which basically live everywhere. They can be found in snow and wastewater treatment plants. And there's the nice classic paper from 2004 where they sequenced the genome of one species of purple non-sulfur bacteria, and they could show with the genome things that they already knew from doing lab experiments, but they could show how it does it. That in the presence of sunlight, it can be photosynthetic. It can get energy from sunlight, but if there are any organic molecules around, it can also use those. So it can be photoheterotrophic or it can be photoautotrophic. If there aren't any organic molecules around, it can fix CO2. So it can be a phototroph, and it can also be either a heterotroph or an autotroph, depending on the conditions. Similarly, if there are organic molecules around, if there are enough organic molecules, it can just eat those to get all of its energy, and it does not need any sunlight, in which case it would be a chemoheterotroph. When it does so, it is using oxygen as the electron acceptor, so it's aerobic. And it can even do this without any organic molecules around. If it has enough yummy electron donors like thiosulfate and hydrogen, which are both really, really good electron donors, it can fix CO2 to get all of its required carbon in the absence of light, in which case it is a chemoautotroph. So this same organism, exactly the same species, different cells in the same population, you expose them to different conditions, and they have the metabolic capability to either be chemoheterotrophs, chemoautotrophs, photoheterotrophs, or photoautotrophs. So I thought that's a really nice demonstration of how amazingly versatile microbes can be. So not only can different species be different from each other, and the diversity of microbial metabolism is visible among the multiple species of microbes, but also just within a single species, you can have a diversity of metabolic pathways, much greater than you would ever see within a single eukaryotic species, for example. And if you really like terminology, there's yet another category that you can insert into this terminology. So we've been talking about aerobic versus anaerobic. We've been talking about photo versus chemo and auto versus hetero. The photo versus chemo is the energy source, that is whether you use light or chemicals, and the carbon source is the auto versus hetero. But you can also specify litho or organo to specify the electron donor. So if your electron donor is an organic molecule and you're a heterotroph, then you are a chemo-organo-heterotroph. So we are chemo-organo-heterotrophs. Normally we just say heterotroph, or sometimes we'll say chemo-heterotroph, but if you really want to, you can say chemo-organo-heterotroph to specify that we are not chemolitho-heterotrophs, where we would be getting electrons from some kind of inorganic molecule like iron or hydrogen, which we would never do, but there are organisms that would do such things. Methanogens, for example, would be specifically chemolitho-autotrophs because their electrons are coming from hydrogen, not from an organic molecule. So here are some fun examples of all of these if you want to go through them. There are some of these that may not exist, and that's a topic for another time, and I can answer questions about it. There we go. That's my very quick overview of microbial metabolism. See you next time.